Welcome to Unit 4, Lesson 5. This will be our last lesson for this unit on genetics. We'll be talking about biotechnology, how it's used, the applications for it, and also the ethics of using it. This is going to be where technology and genetics actually meet together. So when we define biotechnology, this is the application of a technological process, invention, or method that's applied to living organisms. Again, that's where that bio, which means we mean that prefix means living, again connects to the technology part, which technology just really means as something that is used to be helpful. It doesn't have to necessarily be like electronic or something like that. Although we do typically tend to think of biotechnology as something that is super expensive, high powered, a lot of lab sort of type experiences, in actuality, we've been doing biotechnology for thousands of years when we've been selectively breeding domesticated animals and plants. So let's talk about that early and even current to this day type of biotechnology, which we call selective breeding. So, selective breeding is where humans are breeding organisms for specific traits by taking advantage of the naturally occurring genetic variations. In other words, they see a trait that they like in a specific individual. They want that trait in the population, so they want to breed that organism a lot to get that trait into the population. We see this a lot with dog breeds. Dog breeds are the same species, but there are so many different traits that have been highlighted in specific breeds. We see this in corn, potatoes, a lot of other farm crops, ornamental plants that we think things that look pretty, stuff like that. Again, as I said, farm animals and crops are also important. If you look down at this picture down below on the right, you'll even see that we have a lot of plants that we have selectively bred to create new species of crops that we eat to this day. Now let's talk about one of the most earliest selectively bred plants, corn. So back thousands of years ago, we're talking like 6,000 years ago, the Native Americans found this plant in the area of where Mexico is today called teosinte. And teosinte, again, this is the plant that we can see on the left. They bred it multiple times to eventually turn it into the corn plant that we know of today. And this is just one example of selectively breeding that has occurred all over the world, thousands of years ago. And again, as I said before, occurring today. Now, there are some methods to selectively breed. One of them is called hybridization. This method produces offspring with the characteristics of both parents. Typically, it's used to cross very different individuals that have very different, again, characteristics. They're the same species, but they look and have different traits that are very differing. And the idea is that the offspring would have the best of both. The other option is inbreeding. Once you've got this organism that has like the super traits that you want, you don't want those traits to disappear. So what they do is that they will actually breed organisms that are very closely related to make sure that those desired characteristics stay in that line. In other words, this is the action. We see this when we have purebred dogs that are only bred with other of the same breed versus any breed out there. Now, there is a way of increasing variation with biotechnology. First off, you can breed organisms outside of the gene pool with new traits. So you find a new trait, you bring that new trait into the pool by breeding them with organisms in that population. You also can induce mutations through radiation and chemicals. We see this very frequently with bacterial mutations through radiation. And also there's this term called polyploid plants. These are plants that have undergone drug treatments that then lead to a creation of an extra set or extra sets of chromosomes rather than just the diploid that we typically know of. And these organisms, these polyploid plants, typically are stronger and even larger than their relatives. There are a couple processes for genetic engineering. First, you've got to locate the gene and then predict the mRNA sequence that's going to give you that gene. And then you've got to be able to screen a genetic library to find this match. And this is not easy. It's like finding a needle in a haystack, but the technology has gotten better. And scientists have been able to really narrow down to be able to highlight certain genes and locate them through various technologies. One of the things that they do to help them with studying genes is sometimes when you have a sample, there's just not a lot of DNA that you can then study. So there's this process called polymerase chain reaction, or PCR, which actually will multiply the amount of DNA so that you have more to study and more to look at and more to observe and compare. Um, Again, this is specifically used when there's just not enough DNA to then get us enough to be able to study it and to be able to see it in our observations and experiments. This is going to specifically use primers, which are a single strand of DNA, to bind to the DNA sample, which then again makes it easy for us to again handle. We also recombine DNA. So in other words, we take DNA from different sources and join them together. As we can see, we have some splicing of that recombinant DNA happening in the left-hand corner. We typically do this also a lot with plasmids, which we're going to talk about in just a little bit, with circular sort of DNA from bacteria that helps us combine and add DNA to organisms. Now let's talk about using plasmids. Plasmids are small circular pieces of DNA that typically are found in bacteria, but they also can be found in other, usually single unicellular celled organisms like yeast or even other eukaryotes. DNA in bacteria is not the same as DNA in eukaryotes. Prokaryotes, again, bacteria, have circular DNA, which actually is easier for us to modify than our DNA, which is much more complex. So we have a lot of history and a lot of practice in recombining and utilizing plasmids to modify bacterial DNA to then add to other organisms. Now, there is a downside to recombined DNA, which means that you have to have DNA that exists from another source to then insert it. Over time, a development called CRISPR, this technology has been invented to allow us to sort of skip that whole process. In other words, we can edit DNA without having to take it from an alternative source. And this technology has a lot of potential. We're still discovering and utilizing it. There's still a lot of rules and legislation that we're having to follow with it. But this technology has the potential to re-engineer genes and to perform new functions, and also to help prevent disease-causing genes from activating. Now, there's some other terms that we need to know with biotechnology. We have learned how to create transgenic organisms, which these are organisms that have genes from other species. In other words, plants that have maybe genes from other types of plants that have been sort of embedded in there. We also have transgenic animals. We see this with our glowfish. We've embedded glowing genes into these fish that don't typically glow. Now, there's another vocab word that we need to know, which is cloning. And this is where a member of a population of genetically identical cells is produced from a single cell. In other words, we take a cell from an adult individual and cause it to go through cellular division so that we then create a genetically identical organism. This is so much easier if we are cloning unicellular organisms. That's not difficult. But when we get to multicellular organisms, we still have a lot to learn, and there's still a lot of problems with it. Now let's talk about the applications of biotechnology. We have talked about a little bit of this in our research for our PA launch, but first off, agriculture and industry. GMO crops and animals, they have a lot of, again, usage, not only with bringing down prices of food, energy resources, utilizing less herbicides and other kinds of things, and also food scarcity. We also utilize biotechnology with health and medicine. For example, disease prevention. There's a picture of this mosquito. We talked about how GMOs, specifically mosquito GMO, has been developed so that it's not going to reproduce as quickly because the males will die off. Um, we have genetic testing that we use to help learn about people's background and any kind of harmful things that they may have in their genes. We also have learned to sort of help predict genes that they might need to, know about in the future. We also have medical research that we've been doing to, again, help with preventing diseases by medical testing and other kind of uh, experiments that have been done utilizing this technology. Lastly, forensic science. We've been utilizing DNA fingerprinting for a while, where we have DNA that's found on a crime scene and that we're able to match it to DNA that when we're trying to convict or figure out what was going on with a crime scene. Now, there are some ethics and impacts of biotechnology. Anytime that we are involved in something, there are morality sort of rules in place, ethics involved on whether we should or should not do something. And when we can't agree on things, but the universal sort of idea of it, laws sometimes and legislations need to be put in place so that people don't break these ethical understandings. So one of the things we have to look at is the profits and privacy involving biotechnology. Patents. Patents are protections that when someone invents or comes up with a method, or in this case, even a brand new type of gene, that they can put a protection so that other people can't use their idea. Now, this has to be new genes in this case with biotechnology. It can't be something that can be found naturally occurring. So genes in nature cannot be patented. Now, we also need to talk about, again, genetic privacy. We're still trying to figure out what rules and legislation need to be involved in this. DNA can reveal private information, including ethnic heritage, the chances of developing certain diseases, and evidence for criminal cases. And we have these questions. Who owns the information stored in your DNA? And how should genetic information be used? And a lot of people have their genetic information out there. They've given it to different companies for a variety of reasons. Maybe they want to find more about their health. Maybe they want to find more about their family or the historical sort of their ethnicity, their ancestry. There's a lot of that. But again, we have to ask, is your information protected? How is it protected? Who has it? And how can we make sure that it doesn't get into the wrong hands? Now, we're going to end this set of notes with talking about our GMOs. Specifically, we're focusing on GMO foods because that's what people have the biggest thoughts about. And we have some arguments for it and also arguments against it. So, a couple things for GMO foods is that it does produce higher yields. In other words, we can make more because of the efficiency of the plant, the genes, they're better. They're better at doing their thing because, again, we've chosen, the scientists have specifically chosen genes that will make them more successful. This is going to allow a reduce of land use. This also, we see a decrease in energy consumption. The costs of food are lower because there's more food available. We'll have a reduce of chemical usage because you can breed and have organisms that are protected against things that normally we might have to have herbicides or other kind of insecticides or other kind of things to, again, or even like health issues that we would try to, again, prevent by giving them chemicals or introducing chemicals into the environment. We don't have to do that. One of the biggest things is that we've been doing GMOs in the United States for over 2 decades. And there's still a lack of evidence to support that there is harm in this. It's not saying that there isn't. It's just at this point in time, there's not a lot of evidence supporting it. And that's a big argument for people who are against GMO foods. So let's talk about the more arguments against GM foods. Although there is not a lot of evidence supporting harm, There aren't a lot of long-term studies that have been done on this. So people who've had the time to sit down and run studies for years studying what's exactly happening and could this possibly be harmful? We could possibly see insect resistant genes that could threaten beneficial insects. In other words, yeah, you've got a plant that's insect resistant, but now we can involve the beneficial insects, which sometimes are very much involved in that's their habitat, and we need them for our environment, or even with pollination and other kind of things. We could possibly see an herbicide resistant genes that could lead to overuse of herbicides. In other words, um, because we have these herbicide resistant genes, we may need to then turn around and use more herbicides to counterbalance that. And lastly, there is a cost issue when you have biotechnology that has been patented. If you have a small farmer, they may not be able to actually purchase the usage of maybe these GMO crop seeds. They can't afford it, and only big businesses can. Now, that's not necessarily always a thing. There's a lot of GMO crops that actually have been, the patents have been purposefully done away with so that there is a zero cost or minimal cost to small farmers. But mainly, our life is run by businesses and money, and people want that money. So there's still a lot of really awesome plants that are patented that people can't get because they cost too much. All right, that ends our Unit 4 Lesson 5 notes on biotechnology. Please make sure that you have all of this written down. We're going to be going over it the next couple of days in class and linking it to our overall launch of GMOs and what do we know about them. All right, have a great day.